Here we'll learn about thyroid tumors with epithelial origins. To begin, start a table and denote that thyroid tumors can be benign, adenomas, or malignant, carcinomas. Capsular and vascular invasion are key indicators of malignancy. To note that thyroid cancer is the most common form of endocrine cancer, it's in the top 10 most common of all cancers. The most common demographic for thyroid tumors are women over 40 years old. Pay attention that we'll be talking about non-cancerous thyroid tumors, thyroid adenomas, which typically present as nodules and are very common, and also thyroid cancers, carcinomas. The majority of thyroid tumors are sporadic, but key risk factors include pre-existing thyroid disease and/or goiter, exposure to radiation, including X-rays and gamma rays, and iodine deficiency. An important exception to this is papillary thyroid cancer, which paradoxically is associated with too much iodine. Early and late driver mutations are also predictive of cancer and help us link precursor tumors, adenomas, with progression to specific carcinomas. Because thyroid tumors are more common in women, we suspect there's an as of yet unidentified hormonal association. In suspected malignancy, tumors are removed and may require adjuvant therapy such as radiation. Let's review thyroid gland anatomy now. Draw the head and neck in lateral view, then show the esophagus with the larynx and trachea anteriorly. Show that the lobes of the thyroid gland wrap around the trachea. Importantly, thyroid tumors can compress these structures and the associated nerves and vessels and cause hoarseness and difficulty breathing and swallowing. To begin, let's learn about thyroid tumors that arise from follicular cells which produce thyroid hormone. Start with benign tumors, which are non-invasive. These are non-cancerous and are very common, far more common than thyroid carcinomas. Indicate that follicular thyroid adenomas comprise a capsule that has a thin to medium thickness and potentially visible blood vessels. The adenoma follicular architectural pattern varies. Here we show a microfollicular pattern of follicles surrounding small pools of colloid. A single tumor may possess multiple different architectural types. See our notes for more examples. Indicate that follicular adenomas are associated with specific point mutations, which can make them precursors for follicular thyroid carcinomas. Next, move on to Hürthle cell adenomas, which mostly comprise oncocytic cells but are similar to follicular adenomas. Show that the oncocytic cells are polygonal with eosinophilic granular cytoplasm due to a large number of accumulated mitochondria. They have dark, centrally located nuclei with prominent nucleoli. From a genetic standpoint, they may possess point mutations and high somatic copy number alterations. Hürthle cell adenomas often develop into Hürthle cell carcinomas, which we'll address later. Next, let's learn some borderline thyroid tumors. First, indicate that non-invasive follicular neoplasm with papillary-like nuclear features, NIFTP, tumors have a low potential for malignancy, but do have features associated with papillary thyroid carcinoma. These tumors are encapsulated or have clear demarcation, a follicular growth pattern, and nuclear features of papillary carcinoma. Show that nuclear features of papillary carcinoma can be broken down into three key categories: enlarged, elongated, jumbled, or crowded nuclei, nuclear membrane irregularities, such as irregular contours, grooves, or pseudo-inclusions. These look like clear bubbles. Glassy-looking nuclei from chromatin clearing with margination. We indicate their genetic mutations for reference and address additional borderline thyroid tumors in our notes. Now let's switch to malignant thyroid tumors, which are by definition invasive. We'll start with papillary thyroid carcinoma. It accounts for 85% of all thyroid carcinomas, thyroid cancers. Again, this does not include thyroid adenomas, which are non-cancerous. Indicate that these tumors are characterized by finger-like projections of branching papillae comprised of a fibrovascular core and epithelial covering. show the papillary nuclear features, as we drew in NIFTP large jumbled nuclei, irregular nuclear membranes, nuclear grooves, nuclear pseudo-inclusions, and chromatin clearing, sometimes called orphan Annie eye. Additionally, show psammoma bodies between the papillae. These purple-black glassy swirls are unique to papillary thyroid carcinoma and appear to reflect layers of calcium deposited around a small group of necrotic tumor cells. Lastly, show that tumor fibrosis is common. The key distinguishing features of papillary thyroid carcinoma are the nuclear features, psammoma bodies, irregular tumor border, and genetics. Papillary thyroid carcinoma is associated with point mutations. But be aware that the cell architecture may also include classic, follicular, and other variants. These tumors grow slowly and metastasize to nearby lymph nodes. Next, indicate follicular thyroid carcinoma. It's the second most common thyroid cancer. It accounts for roughly 10% of cases. The presence of capsular or vascular invasiveness is key to designating follicular adenoma versus carcinoma. Their cell architecture is otherwise very similar. Let's address several variants of follicular thyroid carcinoma now. Minimally invasive tumors will fully penetrate the capsule. Look for various shapes, including mushroom and hooks of follicular cells growing into the capsule. Encapsulated angioinvasive means that blood vessels have been invaded. The tumor itself is attached to the vessel wall and or covered by a layer of endothelial cells. Widely invasive follicular thyroid carcinoma comprises non-encapsulated tumors that extend into the thyroid and even extrathyroidal tissues. Indicate that follicular thyroid carcinomas are associated with specific point mutations. They spread into the blood vessels and are prone to metastasis into the lungs and bone. Next, indicate that Hürthle cell carcinomas are similar to follicular thyroid carcinomas, but with at least 75% oncocytic cells. Hürthle cell carcinomas are relatively aggressive, with more metastases to the lungs, lymph, and bones. These carcinomas are more common in women, but when they occur in men, they seem to have a worse prognosis. These tumors are associated with a high number of somatic mutations in mitochondrial DNA. The carcinomas we've learned so far are well differentiated, that is, they look similar to normal thyroid cells and tend to grow slowly. Poorly differentiated thyroid tumors represent the next step on a continuum. Right, that these tumors are rare and can arise via de-differentiation of other tumors or de novo. Mutations in TP53 or TERT are later driver mutations that fuel de-differentiation. Cell architecture varies. Here we show the insular pattern in which nests of cells are separated by interweaving fibrovascular stroma. The nuclei vary and can be small and round or elongated, or even convoluted. Tumor necrosis and high mitotic activity are common. These tumors are aggressive and often extend beyond the thyroid. Vascular invasion and metastasis to local lymph nodes or distant sites is common. Tumors may comprise both well-differentiated tissue and poorly differentiated tissue. Lastly, in the continuum, indicate that anaplastic or undifferentiated thyroid cancer completes the continuum. This is a rare but highly aggressive form with a 90% mortality rate. Masses grow quickly and invade local tissues of the neck and throat in most patients, with metastasis to the lungs, brain, and bones. They most commonly occur in women 60 years or older. Like poorly differentiated carcinoma, anaplastic carcinoma is associated with mutations in TP53 and TERT genes. They're also associated with a high number of somatic copy alterations. Finally, let's review another type of thyroid tumor that's derived from epithelial cells, medullary carcinoma. These tumors arise from the C cells, also known as the parafollicular cells, which are neuroendocrine cells that lie outside of the thyroid follicles. Medullary carcinoma is rare. It's characterized by nests of cells with salt and pepper or stippled chromatin. These small groups of cells are surrounded by fibrovascular stroma, and amyloid deposits are commonly seen. Architectural patterns vary. We've shown round ones here. Medullary carcinoma is often sporadic, but inherited forms are associated with MEN2A and MEN2B syndromes and with germline mutations, indicate that C-cell hyperplasia is a precursor to medullary carcinoma and that calcitonin levels may be elevated. Medullary carcinoma spreads via lymphatic and vascular routes. Metastasis to the lymph nodes is common, and the lungs, liver, bone, and brain are the most common sites of distant metastases. This concludes our diagram.